Okay, so I think it's 9 o'clock. So I think we're gonna get started. So hello everyone. Again, I want to thank everyone to join us for today's Immune Zoom seminar. It's again a weekend, so special thank to our speaker. And then yeah, she's really saving my day. We have a couple of speakers have excellent, but Wei's kind enough to jump in and then take the place to deliver the lecture today for the weekend. So it's my really pleasure to welcome today's speaker, Dr. Wei Wu from Lab of Molecular Biology from Center for Excellence in Molecular Cell Sciences, Chinese Academy of Science. I think Wei currently at Neural API just started his lab probably last year. So she's also a trainee from NIH as well. Really happy she's doing well and then be here with us today. So Wei received her undergraduate study degree in bioinformatics at Huazhong University of Science and Technology, and then further to pursue her PhD at the Institute of Biophysics of the Chinese Academy of Sciences, mainly studying the functional roles of long non-coding RNAs in tumors and data integration. After which she then further to pursue her postdoc training. As I just mentioned, she moved to US, joined NCI with Dr. Andrew Nosenswag, who's actually really famous biologist, genetics biologist and then focusing studying deletion and regulation of DNA damage and repair. So from 2022, just last year, Wei actually started her own lab at the Center for Excellence in Molecular Cell Science, Chinese Science Academy of Science in Shanghai, and then focused on exploring machineries of endogenous DNA damage and repair by developing different novel sequencing technologies and bioinformatics workflows. So actually, Wei did a really a productive PhD and then postdoc training. She received NIH Fellow Award for Research Excellence, NCI Directors Intramural Innovation Awards. And I think I read a couple of work from Wei's postdoc training. Particularly, I'm impressed with her study on neuronal DNA damage study, I think, which she's going to talk about today with us. So I think without further ado, Wei, thanks so much. And then we're looking forward to your talk.
Thank you. Let me share my screen. Could you see my screen?
Yeah.
Okay.
Yeah.
So, okay. So good morning, everyone. So first, I want to thank for Dr. Chuan Wu's invitation, and it's my great pleasure to have this opportunity to introduce my work during postdoc training. And this is mainly about program lesions in neuron and its impact during chemotherapy. Because it's immune seminar, so I would also talk about the same program lesions in macrophage as well. So first, I want to explain why we are interested in neurons. Because the brain is very special. It has the highest oxygen consumption to support the high energy demand. And this high consumption of oxygen will release free radicals and result in oxidative stress and also DNA damages. We also try to test the DNA damage signal in iPSC-derived neurons. Without any treatments, we can see very robust DNA damage signal. And also because the neurons are the longest living cells in our body, if the DNA damage are not repaired properly, it may accumulate mutations with age and eventually cause neurological disorders. People have mapped the mutations in neurons by single-cell sequencing, and they found that in individual neurons, it can have hundreds to thousands of mutations, even in very young, healthy individuals. And the number of mutations increase with age. And interestingly, these mutations are not randomly distributed. It's mainly enriched at open accessible region and epigenetic markers for regulatory elements, like H3K4 trimethylation for promoter region, and also H3K4 monomethylation for enhancer regions. And more recently, the same group have mapped the mutations in Austin spectral disorder patients and found serum mutations are specifically enriched at active enhancers. Let me change the laser. But why is this mutation in neuron systems specific at enhancers? It remains unclear, and its knowledge about the DNA damage and repair mechanism in neuron system is very limited. So we are interested into starting the DNA damage and repair in the neuron system. And to study this, we used a system from our collaborator, Michael Werth's lab. She can differentiate iPSC cells to neuron cells rapidly by inducing the transcription factor NGN2's expression. And at day 7, you can see these cells already have neuron cells morphology and can express pineural markers. Now we have cells, then we start to think, how can we map the lesions in neuron? As there are so many different types of endogenous DNA damages, for example, like cytosine demethylation and also ABC sites, 8-oxyguanine, single-strand breaks and double-strand breaks. The frequency of different endogenous lesions are quite different, and also they are repaired by different repair pathways. How can we map the DNA lesions before we know the type of DNA lesions? But luckily, the repair pathways for different lesions has some common steps. First, when there is a damaged base, cell will try to cleave it, and then resynthesize the gap region, followed by ligation to complete the whole repair process. And because neuron already stopped proliferation, so it doesn't have DNA replication process, we can probe the DNA repair synthesis by looking at the newly synthesized region. Based on this, we developed a sequencing technology called SAR-seq. To do SAR-seq, we first incubate the neuron cells with our semi-analog EDU to label the sites of DNA repair synthesis, and then tagged by biotin through click reaction, shared by sonication and enriched by beads. After application and library prep and the next generation sequencing, we will be able to see the location of repair synthesis. If the repair synthesis is not randomly distributed, we will be able to see peaks like CHIP6. After we preferred our SAR-seq assay in the iPSC-derived neuron, we do be able to see many sharp peaks across the genome. And because the mutation in neuron system are at regulatory elements, so we also preferred a tactic to look at the accessible region. And interestingly, we saw our repair hotspots colocalized with the ataxic signal, but not at the promoter regions. So we were thinking, if it's not promoter, are the enhancers? To convert that, we prefer several triptychs for enhancer markers, H3K4 monomethylation, K27 acetylation, and the monomethyltransferase, MML4. And you can see their signal also colocalized with our repair synthesis hotspots. And not just location are colocalized, the intensity between the repair synthesis and the enhancer markers are also positively correlated. But the hotspots of repair could caused by two possibilities. So one is the enhancer region are the damaged hotspots. Like if the damage prefer to happen at enhancers, of course, we will see repair also happen at the enhancers. But it also could be damage occur everywhere. But for some reason, this enhancer region repaired better, so we can see the SAR-seq signal. To test that, we decide to directly probe the DNA damage signal. And when there is a DNA lesions in the genome, the PAP protein will be recruited as the first responder, and then be activated to parallate the other proteins involved in different repair pathways. So we prefer ChIP-seq to parallation. Also, the signal is a little noisy, but you can see some peaks, and these peaks also at our SAR-seq locations. And also the intensities before parallation and SAR-seq are correlated. So now we know neuronal enhancers are hotspots of DNA damage and repair synthesis. Then we start to ask, what's the type of lesions we saw at neuronal enhancers? Are they double-strand breaks or single-strand breaks? So we first test the markers for DNA double-strand breaks, gamma-2X by IF. You can see in the non-treated samples, we don't see any gamma-2X signal. But when we, as a positive control, when we treat the neuron cells with topoisomerase 2 inhibitor at top side, which will generate double-strand breaks, you can see robust gamma-2X signal. And also because our lab has a sequencing assay and seq to detect double-strand breaks, we also preferred our unseq assay, and we don't see any signal at the SAR-seq sites. So we exclude the possibility of double-strand breaks. Then are they single-strand breaks? So when there is a single-strand breaks, like I mentioned before, PAP protein will be recruited first, and then it will recruit another single-strand break repair protein, XRCC1. XRCC1 will act as a scaffold to interact with end processing proteins, DNA polymerase and the ligase to complete the whole single-strand break repair process. So we think XRCC1 could be a nice marker for single-strand break repair. So we also preferred ChIP-seq assay for XRCC1. And now you can see XRCC1 actually at the SAR-seq sites, and also the intensity are positively correlated. But no matter the ChIP-seq for parallation or XRCC1 and also our SAR-seq assay, the resolution is limited by the fragment length after sonication, which is 100 to 100 base pair. But can we directly map the DNA single-strand breaks at single nucleotide resolution? So we then develop another sequencing assay called DDN-S1-seq in order to precisely know which base is broken. Because single-strand break repair very fastly, so we incubate the neuron cells with dideoxynucleotide first to block the single-strand break repair. So the DDN NTP compared to the regular DDN, it doesn't have 3 OH group, so it cannot ligand to another nucleotide. So during DNA repair synthesis, if DDN get incorporated into the neuronal enhancers, then it will stop the polymerization and makes the single-strand break detectable for our unseq assay. And then we embed the neuron cells into an agarose plug and treat the cells with our single-strand specific nuclease S1 to convert single-strand breaks to double-strand breaks. After that, we blunted the break N by XO7 and XOT, and then ligand our hairpin adapter to enrich the broken sites followed by sequencing. If there is a typical double-strand breaks, you will see a pattern like this with plus strand signal at the right side of the break and the minus strand signal at the left side of the break. And you can know the location of the damaged base by looking at the first nucleotide. So we preferred the DDN as one seq in the neurons. And now you can see many single-strand break peaks and see are actually at the SAR-seq sites. And if we look at individual SAR-seq sites more carefully, you can see it's actually not one single-strand break, it's a cluster of single-strand breaks. For example, like cis1 is a single strand break, cis1 is another one, and cis1 is another one. Because DDN S1-seq is a single nucleotide resolution assay, then we also look more deeply at the locations. We found these single strand breaks are mainly at cytosines, and consistent with this, we separate the DDN to DDC, DDA, DDG, and DDT. And you can see only DDC can generate the single strand breaks, but not the other DDNs. Then we start to explore the repair pathways for the single strand breaks. Are they repaired by the single strand break repair pathway I just mentioned before? So in order to test that, we decide to knock down or inhibit key proteins involved in these single strand break repair pathways. So we first inhibit PARP protein's activity by PARP inhibitor. But surprisingly, it doesn't inhibit the repair synthesis, but it actually increases the repair signal. And also when we knock down PARP proteins, we also see an increase of the repair signal. And it's the same when we knock down the scaffold protein XRCC1, we also see an increase of SAR-seq signal. And also it's the same when we knock down the DNA polymerase Pol beta, we see an increase of repair synthesis. And it's the same when we knock down the ligands, ligand 3, we also see an increase of repair synthesis. This makes us very surprised and try to think whether there is alternative pathways. So then we look into the single strand break repair pathway more carefully, and it actually has two sub-pathways based on the length of repair patch. One is the short patch repair pathway, which only replaces the damaged base. And the other one is the long patch repair pathway, which generally a longer repair patch and involved strand displacement. Because by DDN S1-seq, we already know the single strand breaks are at cytosine, and the EDU is a thymidine analog. It actually cannot be captured if by SAR-seq if the single strand break is repaired by short patch repair pathway. But when it's repaired by long patch repair pathway, it's possibly will incorporate EDU and it can be detected by our SAR-seq assay. So when we knock down the key proteins involved in short patch repair pathway, cell cannot repair single strand breaks by short patch repair pathway now. And also single strand break breaks need to be repaired by the long patch repair pathway, which therefore increases the SAR-seq signal. So here we convert that neurons utilize both short patch and a long patch repair pathway to repair the single strand breaks at neuronal enhancers. To summarize the first part, we developed two sequencing assays, SAR-seq and DDN S1-seq, and found neuronal enhancers as the hotspots of single strand breaks and repair synthesis. And this single strand break repaired by both short patch and long patch repair pathway. And then we start to ask the next question, are these single strand breaks at enhancers are only in neurons? Can we see the same single strand breaks in the other systems? So we choose that system because the lab already has it. This system is from pre-B to transdifferentiate to macrophage by inducing the expression of CBP alpha. And the cells can be transdifferentiated to macrophage rapidly within 2 days. And you can see here at day 2, most of cells already become macrophage. And then we preferred our SAR-seq assay. After inducing the transdifferentiation 1 day, we add the EDU to label the sites of repair synthesis. And then after 1 day, we collect the cells for SAR-seq library prep. But not surprisingly, we didn't see any peaks. So it could be caused by 2 reasons. One, there is no single strand break at enhancers, so we didn't detect anything. But the other one is maybe it's repaired by short patch repair pathway, so our SAR-seq cannot detect it. So we decide to test the 2 possibilities by prefer DDC S1-seq to directly map the single-strand breaks, and also prefer SAR-seq by inhibiting short patch repair pathways. So when we add EDU, we also add PARP inhibitor to inhibit the short patch repair pathway, and then do our SAR-seq assay. And now you can see we can get a lot of peaks at the genome after PARP inhibition. And also when we did our DDC S1-seq, we can also see many single-strand breaks at these SAR-seq sites. And these single-strand break signals are also at macrophage enhancers, and the intensity also very positively correlated. Then we start to ask, what could be the source of the enhancer single-strand breaks? Between these two systems, the common feature is it has to change the transcriptome from iPSC to neuron or from pre-B to macrophage. And an important step to make this transcriptome change is you have to activate the important regulatory elements like enhancers by DNA demethylation. And actually one of the DNA demethylation pathway involved single-strand break generation and repair. To briefly introduce the DNA demethylation pathways, so normally the cytosine can be methylated by DMT proteins to 5-methylcytosine, and the 5-methylcytosine can be further oxidized by TET proteins to 5-hydroxymethylcytosine and 5-formylcytosine and carboxycytosine. And in dividing cells, there is a replication-dependent dilution pathway called passive demethylation. And in non-dividing cells, the active demethylation mainly addressed by the active demethylation, which is the 5-FC and the CEC can be cleaved by a glycosylase called TDG and repaired by single-strand breaks to put our unmethylated cytosine back. So in order to track whether the TDG-dependent active demethylation is the source of the single-strand breaks, we first check the TET protein expression in both systems. In iPSC to neuron system, you can see TET2 and TET3 proteins are stimulated. And also from pre-B to macrophage system, you can see TET2 expressions are activated. And also we preferred sequencing assay to map the 5-FC and 5-HMC in neuron system. We also see a nice correlation between these active demethylation intermediates and our single-strand breaks. But we want to more directly test whether active demethylation is the source of the endogenous damages. So we knocked out the TDG protein in some pre-B to macrophage system. And you can see, luckily, after TDG knockout, the pre-B still can be differentiated to macrophage. So we can prefer our SAR-seq and DDCS1-seq assay. Also, the cells still can be transdifferentiated to macrophage, but you can see there is no single-strand break and the repair synthesis signal after knockout TDG, which indicates the neuronal enhancer, which indicates the single-strand breaks at enhancers are actually caused by active demethylation. So we also try to generate TDG depletion in the neuron system. We create a detect inducible TDG, detect inducible knockdown system for TDG. To do this, we knocked in our FKPB tag into the N-terminal of TDG gene. And then after 6 days of differentiation, we add detect to induce the TDG protein degradation. We first checked the DNA damage signals correlation by F, and you can see in the non-treated samples, we can see very strong DNA damage signal. But after we knock down TDG, the correlation signal dramatically reduced. And also, if we detect single-strand breaks before and after TDG depletion, you can also see the single-strand breaks are gone after knockdown TDG, which also indicates the neuronal enhancers single-strand break are caused by active demethylation. So to summarize the second part, we conferred the cells of the single-strand breaks at enhancers are from the TET protein TDG-dependent active demethylation. And in neuron system, it's repaired by both short patch and long patch repair pathway. But in macrophage, it's only used the short patch repair pathway. Then we start to think, because normally these endogenous lesions are very well controlled by cells and will not lead to any consequence. But what happens if there is additional treatments, for example, like chemotherapy? So chemotherapy is the mainstream treatments for cancer patients, but sometimes the chemotherapeutic agents can pass the blood-brain barrier and cause neurological toxicity, which is also known as chemobrain phenomenon. Chemobrain is one of the most common side effects in cancer treatments. More than half of the patients will report different types of defects in like concentration, memory, motor functions, and etc. It can be short-term and long-term consequence, but the underlying mechanism remains unclear because most of the chemotherapy agents mainly target the cancer cells because they're rapidly dividing. But how can it affect neuron cells is unclear. And also, earlier this year, the Cancer Grant Challenges supported by NCI and Cancer Research UK released the 9 new challenges in cancer field. And we also found chemotherapy-induced neurological toxicities are one of them, which also indicates it is important to study the mechanism behind it. And to study the mechanism of chemobrain, we first mainly focused on the group of chain-terminating nucleotide analog agents. So these agents kill cancer cells mainly by inhibiting DNA replication because they can be incorporated by replicative polymerase during DNA replication and stop the DNA synthesis. And the 2 most widely used chain-terminating nucleotide analog agents are IRC and gemcitabine. IRC is mainly used in the treatment of leukemia, and gemcitabine has been used in different types of solid tumors. Both of them are cytosine analog and has modified base. But the question is why they will affect neurons. Also, neurons don't have DNA replication, but as we already showed before, it has very active repair synthesis at the enhancers. It is possible that these agents can be incorporated by the repair polymerase and generate DNA damages, which could potentially cause neurological toxicity. So in order to test that, we preferred our SAR-seq assay after IRC treatments instead of DDC. So if IRC can be incorporated and generate DNA damages, we will see peak signals at enhancers. And it's true, we can see very sharp single-strand break signals at the neuronal enhancers as well. And interestingly, this incorporation is cytosine analog specific. So if we treat the cells with the adenine analog ARA-A, which has the same base modification as IRC, we don't see the single-strand break signal at the enhancers. And also because if there are 2 single-strand breaks nearby on opposite strands, it may consider as a double-strand break and trigger double-strand break response. So we also checked first the end-seq signal after DDC treatments without S1 cleavage. And we do be able to see some mild double-strand break signal. And also after we do gamma-2X IF, we can also see a strong gamma-2X foci formation after RIC and DDC treatments. And this gamma-2X signal is TDG dependent. If there is no activity methylation since there is no incorporation of RA and DDC, RC and DDC. And it's the same, we also checked another DNA double-strand break marker 15-3BP1. We can also see TDG-dependent 15-3BP1 foci. So we want to know whether these agents does inhibit repair synthesis of the single-strand breaks at enhancers. So we preferred our SAR-seq assay after treat the cells with the different cytosine analog, DDC, RRC, and gemcitabine. But we also didn't see a reduction of the repair synthesis. We actually see an increase of repair synthesis. So we think what is happening is that, So we also tested the increase of the SAR-seq signal is cytosine dependent, like the S1 and 6 signal. So if we treat the cells with adenine analog, DDA and RA, we don't see any increase. But if we treat the cells with cytosine analog, DDC and RRC, we can see increased signal. And we think what is happening is that after active demethylation generate single-strand break, and during the repair synthesis, these agents get incorporated and generate a new DNA damages. And these lesions will be further processed and repaired until it's fully removed. So which in the end increase the repair synthesis signal. In order to track that, we prefer the live cell image of 15-3BP1 foci formation. And you can see 15-3BP1 foci form and then disappeared, which indicates the single-strand breaks are actually repaired. And in the end, we also checked the toxicity caused by RRC. You can see the neuron cells start to die after 1 day treatment of RRC, but when we knock down TDG, it's fully rescued the toxicity. So to summarize the last part, we found during the endogenous single-strand break repair, the chemotherapy agents RRC, gemcitabine can be incorporated during this repair synthesis and cause DNA damages, which could potentially lead to neurological toxicity. And hopefully this result can help to explain one of the mechanisms for chemo brain and help to optimize the therapy design. For example, treat the patients together with TDG inhibitor with RRC, or try to develop the other non-C agents. And in the end, I want to thank my supervisor, Andre Nussensweg, for his supervision and their support for these 2 projects. And also want to thank my colleagues, Dongpen, Elsa, Will, and Jiachen, who did most of the experiments. And also my current lab members who is doing some follow-up of these projects, and also the other collaborators. And thank you for listening. Happy to take questions.